Hello, my name is Jonathan Dorn and I'm a science educator based here in Scotland. The purpose of this video is to provide some explanatory details and demonstrate some of the key practical components of the characterisation of lactic acid bacteria from Kefir activity. The activity has been designed to support the delivery of the National 5 Higher and Advanced Higher Biology qualifications here in Scotland. However, the significance of lactic acid bacteria and their connection to food production in particular might mean that you're able to find a suitable use for this experiment, either in whole or in part, in a range of other subject areas such as the National 5 Health and Food Technology course. The activity has been divided up into 3 stages, with each one being an escalation in both material requirement and complexity. The lab is cumulative, which is to say that in order to undertake stages 2 and stages 3, you must first have completed stage 1. In addition to this video, the file that has been shared with you should contain a very succinct one-page protocol that's designed primarily for your benefit as the teacher. You should also find a 20-slide PowerPoint presentation that I hope you might find helpful in delivering your own lesson prior to the lab, as well as a risk assessment. Kefir is a fermented milk product that dates back thousands of years and was initially deployed as a means of preserving milk in the Caucasian mountain regions. It's made from a mesophilic symbiotic culture of yeasts and bacterial species like Lactobacillus and Acetobacter. It can be an interesting substance to examine with your class when teaching them the principles of microbiology. And although it's always vital to be vigilant when undertaking any practical work, being aware of any diversity in student need, as food-grade organisms, their safety profile is quite appealing for a school setting also. So let's take a look at the lab. In this small flask, I have a Kefir culture that I commenced a little over 24 hours ago by inoculating some sterilised milk with a commercial culture that I purchased from a manufacturer, so I know exactly which organisms are contained within it. I inoculated the milk, sealed the container, and placed it in a warm space for that window of time. And the concoction of Saccharomyces and lactic acid secreting organisms have been busy making this place their home by colonising the milk during that time, and it's now ready for use in our stage 1 practical. The first step of this activity is to label 5 universal bottles as 100%, 75%, 50%, 25% and 0% respectively. If you're anything like me, you'll find that your handwriting degrades quite considerably when confronted with nitrile gloves and curved glass, but as you can see we got there in the end. Following this, the bottles are initially filled with the appropriate volume of milk, with the 100% Kefir culture left empty, and then topped up using the corresponding volume of Kefir culture for a total volume of 20 millilitres each. All that remains now is to allow this symbiotic culture to grow for around 24 to 48 hours. So now that 48 hours have passed, we can examine our cultures and should find the 100% Kefir to be, as in this case, verging on a solid when compared to the milk control. This has primarily resulted from a combination of the secretion of polysaccharide sugars as well as the coagulation of the milk protein casein. I can also confirm, very much confirm, that the production of lactic acid and other volatile aromatic compounds should be quite evident to your students too. In addition to these products, it is possible to demonstrate the release of carbon dioxide very easily by simply placing a suitable Kefir culture into an appropriate container and affixing a balloon to the top. Stage 2 of the activity requires a little more by way of material availability, but also empowers your students to engage in some of the staples of real-world microbiological investigation, in particular the construction of streak plates and the use of selective or characterising agar recipes. As you can see, in this instance I've opted to create a batch of nutrient agar plates which are general purpose and will cultivate a wide range of organisms that do not have particularly complex nutritional needs. And additionally, some McConkey agar plates which will selectively isolate gram-negative organisms and contain neutral red as their indicator, giving rise to pink colonies when the microorganisms in question are fermenting lactose. Although it is very possible that your students will be able to master the lawn plate spreading technique, demonstrated adequately here with minimal guidance, they are likely to require a little more support when constructing their streak plates. A small diagram of the favoured method when attempting to acquire isolated colonies is shown on the teacher's protocol provided, but you may also wish to consider allowing your students to practice the method first using distilled water before attempting it with microorganisms. As an undergraduate, my microbiology instructor would joke about the infamous first-year sizzle, the sound of a red-hot wire loop and the charred bacteria it has been dipped in makes when pressed against agar. You may wish to caution your students on the risks of overheating their loops, or, as I now do, use the sizzle as a teaching opportunity on the thermal limits of microorganisms and the best practice when trying to culture them. In the context of a Kefir culture, the organisms selectively identified via the use of McConkey agar are likely to take the form of gram-positive Zymomonas and Acetobacter species. However, this is an excellent opportunity to get creative with your use of indicator additives. Colleagues and I have had excellent results with plates that have been laced with bromocresol purple, which will yield bright yellow colonies when inoculated with lactic acid secreting bacteria, as well as bromothymol blue, which will yield similar results. Instructors should note that whilst yogurt and kefir organisms typically bear a generally regarded as safe designation, the CERC Safety and Microbiology Code of Practice designates Acetobacter as a level 2 bacterium, and this may have some implications for the deployment of PPE. As you can see from the protocol, stage 3 involves the chance to conduct both sim motility testing and a gram stain. A more detailed video resource describing these protocols is available and includes some suggestions on how these techniques, which are made reference to in both the higher human biology and advanced higher biology specifications, might be best positioned within the curriculum, or even adopted to fulfill project-based coursework requirements that these qualifications entail. I very much hope that you found this video and the accompanying resources to be helpful. Practical activities not only serve the purpose of complementing and deepening the theoretical knowledge of our learners, they can also help us to engage our students and be a great vehicle for relaying our own passions to them. And with that in mind, I hope that both your students and you yourself find this lab to be enjoyable should you choose to undertake it. Thanks for your time.